Rick asked a short while ago about my thoughts on pulpal stem cells, and these cases that I'm doing from reeds have given me some thoughts on what these pulpal mesenchymal stem cells really are like from the imaging point of view. When you look at our classic H&E histological sections, we get a sense of what the odontoblastic layer is like, that it's filled with these odontoblasts and maybe pre-odontoblasts. But we really don't have a very good notion of what that layer actually looks like during reparative dentin or replacement dentin or irritation dentin. And my thinking in looking at Reed's cases is that this layer, this odontoblastic layer, really probably contains the mesenchymal stem layer as well, and it's all jumbled up inside that. Because remember, those mesenchymal stem cells become odontoblasts, and the imaging on those cells, in my literature review, is very different from what I'm seeing when I'm looking at Reed's cases and interpreting as mesenchymal stem cells. So there's a great deal of uncertainty. This is Reed's case 641, and looking in an area where you don't have a lot of the sectioning debris. When you actually go into the stitched images on the lingual canal, and you start at the 30x stitched images, and you go in and you look at this, it appears like the mineralization front is covered by a layer of cells in certain areas. And it's the nature of those cells that is so different. So I don't know if it's the Triton that does this, but when you look at this, you can see there's like a layer of cells right above the mineralization front. And this was a vital case, if you recall. And when you actually go in and look very carefully at those, these cells, they look like white blood cells almost. They're obviously some of the odontoblast cells are there, but my interpretation of these are mesenchymal stem cells. And when you actually go in and look very carefully, it's worthwhile studying these. And you can see some of the odontoblastic processes are still in the tubules, and there you see some predentin, mineralized predentin that's being demineralized. But you can also see some remnants of odontoblastic processes, and what the spindly shaped cells are, unless someone has a better idea, I believe that these are mesenchymal stem cells. If you look throughout the sections, you'll see that it's like a syncytium. They're all connected to each other, right on top of the predentin. And some of them look like they're in the process of becoming odontoblasts. So my interpretation of this, I'm not a pulpal biologist, but there you can see the demineralization of the predentin that masquerades as bacterial morphotypes. And I know Rick and I have discussed this. I'm not ready to call those bacterial morphotypes. I think that's from the demineralization of the predentin, and they can look just like bacteria. So you need to be careful. And if these aren't mesenchymal stem cells, well, then what are they? You go in here, you can see they're gone down into the tubules, perhaps maybe this is an irritation dentin or a replacement dentin area in the root. I don't know. But these are all mixed in with what are clearly odontoblast cells. So it's a very interesting thing because there's nothing in the literature on the effect of Triton irrigation or the methodology that he actually used to instrument these. But I can't imagine that the irrigation converts red blood cells into looking like this. Um, these are definitely cellular in nature, and my interpretation is that these are mesenchymal stem cells. I could be very wrong, but that's what I've surmised by looking through all of these stitched images. Here you see another area in that case where there's a lot more demineralization. All of the peritubular dentin is gone. There you can see some of it being dissolved away as well in stages. And when you go into other areas at much higher power, you do get some areas where there aren't that many stem cells, and they've been pretty much taken care of by the irrigation, but there's a few right there that are still present. Right up against the mineralization front, that darker area is the mineralization front. So in this next case that I'm doing of reeds, I'm going to make a report on it, so I don't want to go into the details of the case, but if you actually go into this case, you see something actually very similar, and this was a Triton case as well. In that area where you see kind of, it looks like maybe a remnant of a blood clot or something, but when you actually go into it, and we're going to do a stitched image in that area, and when you do that, you see a layer that looks a lot like a layer of stem cells. I just can't believe that these are red blood cells. It looks to me like that is a stem cell layer, and this is a completely off the cuff, because there's nothing really in the literature on what the irrigation with Triton does to these cells. A little further down the area, you get a sense there's see, those look like maybe red blood cells, but they're completely covering the mineralization front, and then when they're gone, you're right there at the dentinal wall. This is the other half of the sagittal section, and again, you'll see in my report on the case, the canals were very, very clean. No evidence of any bacteria at all. But when you look at this, that is kind of how I interpret how I thought the stem cells would look. And in the literature, under the SEM in the literature on stem cells, that's kind of what they do look like. They don't look like these spindly shapes. So that gives the appearance of what we've been trained to view as stem cell extensions over the mineralization front. So we're getting a completely different kind of picture of what these cells are. And I may be completely wrong. Maybe these aren't stem cells. But since Rick asks, that's what I'm seeing. And I'll do a report on the latest case of reeds and cover it maybe a little further.